Good afternoon. I, Dhruva Jani, heartily welcome you all to the series of invited talks by our promising alumni, organized by Gujarat University Botanical Society and Gujarat University Learned Alumni of Botany. We have with us our Honorable Vice Chancellor, Professor Himanshu Pandya, as President of Function, our Pro Vice Chancellor, Dr. Jagdish Bhavsar, and our Registrar, Dr. P.M. Patel, as our guests of honor, Professor Bharti Dave, Dean of Indrasil University, as our chief guest, our distinguished resource person, Dr. Rupesh Maurya, Assistant Professor at Bioscience Department, Indrasil University, faculty members of the department, Archana ma'am, Solanki sir, Maitre sir, Modi sir, Soumya sir, and participants. It is indeed a pleasure to have you all with us here today. Gujarat University is on a fast track of progress. Gujarat University has successfully achieved 5-star ranking in GSIRF once again, and it is further selected in top 50 universities in India by NIRF ranking 2021. We are proud of our university, and as a mark of respect, let us rise for our university song.
वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन गुजरात यूनिवर्सिटी आपणी गुजरात यूनिवर्सिटी आपणी विद्यातणु तपोवन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
राष्ट्र चेतना कला साधना खेल भावना विकसे
विश्व मानवी बनी गुजराती विश्व सकल માં વિલસે સ્નેહ સંપ સહકારની સાથે દેતી તું સન્મતિ સૌના સાથને સંગાથે સૌ સિદ્ધ કરે ઉન્નતિ પ્રેમ બંધુતા સમાનતાના પલ પલ પ્રસરે સ્પંદન વંદન અભિનંદન વંદન અભિનંદન वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
Our Honorable Vice Chancellor, Professor Himanshu Pandya, our Pro Vice Chancellor Dr. Jagdish Bhavsar, and our Registrar Dr. P.M. Patel will join us in the webinar soon. Now, we shall move on to further webinar. Our chief guest, Professor Bharti Dave, is here with us. She is role model for all of us. Her teaching and research acumen is beyond comparison. She retired as professor and head Department of Life Science from Bhavnagar University and is now with Indrasil University as Dean, School of Science. Ma'am, we request you to bless us with your words of wisdom.
So I guess meanwhile, Dhruva, let me just formally welcome. Ma'am is joining. I think she'll just take a minute. It's indeed a pleasure to welcome you all here for this series of invited talks by our meritorious alumni organized by Gujarat University Botanical Society and Gujarat University Learned Alumni of Botany. We heartily welcome our Pro Vice Chancellor Sir, our Honorable Vice Chancellor Sir, and our Registrar Sir, who have always supported us and blessed us. We heartily welcome Professor Bharti Dave, our chief guest, who is an embodiment of courage, commitment, and has always inspired us. Ma'am, we are very blessed to have you with us. Thank you so much for joining in time. We welcome our meritorious alumnus,
Thank you very
Dr.
much.
Rupesh Maurya, and are indebted to him for accepting to share his vast knowledge and rich experiences in taxonomy with us. I also welcome my colleagues in the department, Professor Hitesh Solanki, Professor Bharat Maitre, Dr. Nanish Modi, Dr. Soumya Patel, and all the participants. Today, we'll try and reinforce the significance of plant identification, an area of study that is fundamental to all researchers in botany and related disciplines. I once again welcome Rupesh sir and request you sir to share your expertise and help the budding botanists in identifying plants around us. We look forward to a great learning experience. Thank you. And now I request Bharti ma'am to please bestow her blessings on us. Over to you, Bharti ma'am.
Okay, a very good morning to all. Respected Archana Ben, Maitri Sahib, Nainesh Bhai, Hitesh Bhai, and you can say my colleague, the most junior colleague, Rupesh over here, and Dhruva Jani. In fact, I know each one of you. I have worked with, except Archana Ben, I've worked with everybody over here. I have worked closely with Dr. Maitre. I work closely with Hitesh Bhai also. And so, really, it's very nice for me to be here. I've also worked closely with your Honorable Vice Chancellor, Himanshu Bhai Pandya. I have my utmost respect for him. So, it's my pleasure to be with you today, and specifically when my junior colleague, which I treat him as my younger brother, is with me today. Both Nainesh Bhai as well as Hitesh Bhai, both of them had recommended him, a student of your university and your department, to me, and he is really doing very well, and I'm really very fortunate to hear him today in a form of the lecture that he would be delivering. As I saw to his topic, I think the target student is MSc students.
Yes, yes, ma'am.
Yeah. So as this thing, I saw to the topic on which he's speaking, it is plant identification, which you can call it as a part of taxonomy. Previously, just before 3 to 4 years, I used to believe, and even my students where I was working used to believe, okay, taxonomy, they found it to be very, very boring. That sometimes many students used to tell me, okay, it is, madam, a tax on me. So, but now seeing since the last 7 to 8 years, seeing to the importance of taxonomy, it may be microbial system, it may be plant system, it may be animal system. We all know the importance that taxonomy has. Specifically, if I talk about plant taxonomy, it has its importance in each and every field. You talk of any industrial sectors also. Our own parent group, that is Candila, it has almost an entire, you can say, section devoted to herbal drugs. And we have one of the very senior botanists, I think she's an alumni of your university, Dr. Vandaman Modi. She's all alone handling the entire herbal product division on her own. So see to the importance that plant taxonomy has. Without naming a particular organism, without knowing what that particular organism is, what are their characteristics, whatever research we do, it may be at the topmost level, it may be a research at an updated level, but it has no meaning. Many times you also might be experiencing, whenever we take viva of certain students for their PhD, the organism that they work with, all of you might have had that experience. You just ask the characteristics of that particular organism, plant, bacteria, microbes, or animal. They are not at all aware of the fundamental characteristic of that organism. So that is the major drawback on our part. So taxonomy has that particular importance, specifically identification. Any industrial sectors besides the academic sectors that the students opt for after getting their master's degree or PhD degree, each and every industries, it may be pharmaceutical industry, it may be drugs industry, it may be dye industry, it may be the resin industry. All these industries have plant taxonomy or plant identification or herbal drugs as a system, and where the botanists are required the most. So I think, Archana, when the topic that you have asked Rupesh to speak on is very apt, seeing to the present situation.
Thank you, ma'am.
Yeah. So I congratulate you, and it is really a very good initiative where all your alumni are being asked for to deliver that lectures, because the students who would be getting out of their masters or getting out of the department with master's degree or PhD degree, it is very well this thing. They should know who our alumni are. They may be help to any one of the students who are coming out. You never know. We go for interviews and we have our students sitting in front of us. So if the interviewers are alumni, it really makes a very huge difference. It may be government sector, it may be private sector. And I should really tell the students that the field that you have chosen is really very good. If I give you one example of how all, specifically the master's students, should work on is, if I give you an example of Dr. Mashekal, many times in my department where I used to be earlier at Bhavnagar University and where Professor Maitre also was, I used to tell the students during the lectures that I used to give, you must be knowing Dr. Mashilkar, he was the former Director General of CSIR in New Delhi. He always, during his lectures, he always used to give gratitude to the teachers that had taught him. And always one example that he gave was of his physics teacher when he was in school. The physics teacher was Dr. Barwe. And he was explaining to the students what is the focal length. How can you calculate the focal length of a lens? Theoretically, the students were not able to understand. Then he took the students out in the sun in the field. And in the field, he took a piece of paper, he took a convex lens, he put up that piece of paper in front of a convex lens. All the rays from that lens used to merge at one particular point. And within seconds, they had a specific spot on that paper, and the paper started burning. So that he told, okay, the distance between the lens and the spot that you get, in this way you can calculate the focal length. But after saying this, he immediately turned to Dr. Mashilkar, and what he said was, he did not tell the name in front of all the students, but he spoke to him, okay, if you focus your energies like this, you can burn anything in the world. So from this, he said, okay, I got two philosophies from my teacher. One philosophy that he got was, science is so powerful, and from that particular point, he had decided to take up science as a career. And the second, if you focus your energies like this, if you focus your energy towards a goal, you can achieve anything in the world. Sky would be the limit for you all. So I wish to tell all the students that the field that you have chosen is science, and specifically plant biology or botany as you call it, has lots of scope, would make you into a very good educator, would make you a very good scientist. And if you want to enter into academics, it would make you a very good teacher too. So with all this, I congratulate Archana Madam for taking up this very good initiative. Professor Maitre, I'm very delighted to see him. Hitesh Bhai, I cannot see him on the screen. Dr. Nainesh Modi, he's a member of our Board of Studies here in Botany. And with this, I wish the students the very best. Please do not neglect this area of plant identification. Go to the field, be with your teacher in the field, observe what is in the field, think over it, discuss over it, and then you will come with a very good solution. So this is what Dr. Rupesh, being trained by all of you, great teachers, inspiring teachers, is doing this to our students. Regularly, at least once a week, he's taking the students to the field, because it is in the field that science lies. Completely only relying on wet lab experiments would not take you anywhere. You need to observe it in the nature. You need to think. You need to observe what it is. You need to discuss with your teachers and then come to a solution. So with this, my best wishes to all the masters as well as PhD students, and my congratulations to all my colleagues over here whom I know them very well. Thank you very much.
Thank you so much, ma'am.
Thank
Yes,
you,
ma'am.
Bharti ma'am.
Thank you,
Now
madam.
we shall start. Now we shall start with our technical session. We have with us our distinguished resource person, Dr. Rupesh Maurya. Sir is also our alumnus, and we are proud of his achievements. Dr. Rupesh Maurya is an assistant professor at Department of Bioscience, INRISL University, Kadi. He pursued an MSc degree in botany from St. Xavier's College, University of Mumbai. He then completed an MPhil and PhD degree from Department of Botany, School of Science, Gujarat University. He explored diversity and ethnomedicinal traditional knowledge of Panchmahal district of Gujarat as a part of his PhD study. Dr. Rupesh has keen interest in plant diversity and solving taxonomic problems. He was JRF in Biodiversity and Ecological Assessment of Jambugoda Wildlife Sanctuary project funded by Gandhinagar Forest Division. He has worked on the biodiversity of plant in different regions of India with several consultancies such as EcoBasics, Terracon, Magdalene, Almeida Environment Center, Drivishta Consultancies, and NGOs like Shrushti Organics and Poitrabai Foundation. Sir has authored and co authored many scientific papers, technical reports, book chapters in the field of biodiversity and ecology. We are delighted to have you with us, sir. And thank you, sir, for joining us in the series of online invited talk organized by Gubs and Gulam. I request you, sir, to deliver your talk on plant identification, rightly titled Pehchhan Kaun.
Uh, just a minute, Archana, can I
Yes.
leave now as I have a meeting?
Yes. Yes,
Yeah.
yes, ma'am. Thank you so much.
Okay, thank you so much.
Yes, over to you, Rupesh.
Over.
Only. And what I have learned from the department, I'm going to deliver the same things and again to the other colleagues as well as students. And so students, so we need to, so only this recently Bharti madam was talking about the importance of plant identification. And so we will ask some in continuation of that, we will ask some questions like basic questions. So what is biodiversity? So biodiversity is basically variability of the life form in from gene to the ecosystem. And we how many next question suddenly appears, how many species do have, how many kind of species we have on earth? So you can see like there are very variety of plants, animals, bacteria and other living forms are there which are available on earth. And if we estimate the number of these biodiversity, the species, so they will come up to 10 to 14 million. And still many species are waiting to describe, waiting there, we are waiting still to describe. And so the estimated number, like how much we have, so estimated number is around 30 million species present on earth. So still we have described 10 to 14 million, half of the diversity is still waiting to describe it. Then next question appears, like some students ask generally this question, why do we study biodiversity? So for that, the in current perspective I will talk. So we have like lots of industries, as Bharti madam was introducing, like there are like a range of industries which require botanies, which requires the identification skills. And but recently the RUN, they have set the sustainable developmental goals for 2020. They are planning to achieve 17 developmental goals such as no poverty, food security, the fight against hunger, women empowerment. And they requires the knowledge of the biodiversity. These goals, all goals are going to require the knowledge of the biodiversity like for next 10 years. So we need to understand this. So what we are getting from the biodiversity is like very help, is like we need to understand this. We can categorize the ecosystem services into four different parts. So we are getting basically ecosystem services from the biodiversity. We want to categorize it. We want to categorize it in four different kind of ecosystem services: the provisional ecosystem services, regulatory ecosystem services, cultural and support services. So what we are, well, industries are based on like fuel, food, fiber, timber, dye, resin. These kind of ecosystem services are known as provisional ecosystem services. Then we have regulatory. Regulating ecosystem services are basically what is the climate regulation kind of services comes under regulating ecosystem services. Then we have support. The factors necessary for providing such ecosystem services are comes under the support services like soil cycle, nutrition cycle, hydrological cycle. Such services are into the support services. Then we humans have evolved with the nature, and our culture is basically related to the our cultural services, our culture related to with the nature. So these like spiritual, aesthetic value, educational purpose, these things comes under the cultural services. So we are getting almost everything from the biodiversity only. So we need to understand this, and we need to how without identification, the first step is the identification with working with the biodiversity, and we are going to deal with the first step, that is identification. There are various other steps are also, I will not deny that, but first step is the identification. And we need to learn that, like for solving these 17 sustainable goals which are going to affect the millions of the life on the earth. So we have, so we'll ask a basic question now. I will ask basic question. What is the taxonomy? So taxonomy is basically categorized into four different parts. This is the branch of biology which is deals with the nomenclature, identification, description, and classification. And the what is, so we are going to deal today with the nomenclature identification, because we cannot separate these two things. Okay, so in this, the what is identification? The identification is the process and of so showing, recognizing, and giving proof. That is very much important. I would like to highlight the giving proof in the process of the identification. So I will share some important facts. Look at this. So these are some shocking facts. So in this is Netherlands lab. They have what they had. So they have the 3,210 people they have assessed. They have asked a basic question to them around based on the native species identification, and they found out interesting results. The biodiversity professionals, the 89% who there are biodiversity professionals who can identify the good correct identification. And general public, they 75% of the general public, they are failing to identify the native species, and these are the just 27 native species. Okay, so they're not 100 200, just 27 native species, common species they have shown to the local public. And so, and the lack, so there is a lack of species recognition in general public as well as the children's. Only 35% of the children they can identify the native species. So this is a very indicator. This study is basically indicator, and this is worrying also. So we are going away from the nature. So this is indicator. Identification is here. They have used as an indicator how we are going away from the nature. And like you have, and the paper what Nishit lab his Nesbit lab has given, they have taken out of 502 sample of the plants, and what they have find out. So they have find out that only 37 species are basically best practice for the plant identification, and others are not. Like out of 502 plants specimen, only 37 were correctly identified. And so this to the a very good study I would like to quote here for Lucas lab. And what they have the written very nice way what they are writing, plant identification credibility in ethnobotany. So you know that the term ethnobotany is basically documentation of the traditional knowledge what we are getting from the ancestors. So this is very popular subject, and this is going to help us the documentation as for many industries also. This knowledge is going to help us. But the shocking results, out of they have taken more than 45 publications as a from one country. Okay, and what they find out, only 2.3% taxa in the publication were correctly identified. And they even, there are many publications in ethnobotany, they don't have the voucher specimen. So this is basically the what Lucas study is saying. And the voucher specimen was not correctly identified, and there are many places where genus was not correct. Even genus was not correctly identified. So this is the basically worrying things. So what common mistakes? So I have these taken the common mistakes from the like scientific literature. So what common mistakes like our authors are doing in the plant identification? So they are failing in use of the scientific names. You know, like we are the botany students and we want to write how the scientific names for the particular plant in our manuscript, whether if I am working for the embryology, whether I am working for the physiology, or some other field I'm working, allied field I'm working, then still we in our manuscript how to write the scientific name. Many places we have found out like there are misspelling in the scientific name and incorrect author names are seen. So even many places these synonyms in the plant, this is misleading to the authors also, new authors also, and as well as the old authors also. This because while selecting the plants for the particular problem, solving particular problem, well, many times we started using the synonym of the plant. Then we need to understand we are failing to use the taxonomic reference also. So this is a very thing for us. So what is the what is the best practice? So students, we need to have our voucher specimen. What is the voucher specimen? For any kind of study, we need to have our voucher specimen. What is vouchers? It is basically a well-mounted, neatly labeled specimen, dried specimen. So when I say specimen, basically it is herbarium sheet, the properly labeled herbarium sheet, which have different characteristics. It has flowers on the or the fruits, from where we can identify that particular specimen. So the voucher specimen, after making the voucher specimen, we should go for the identification of the voucher specimen. So how to go with that? So either we can send the specimen to some expert, some herbarium, and or you can but for the voucher specimen, we need to have a board with the herbarium, and herbarium only can give a letter for like for your further work. So this is basically the procedure is known as authentication of the specimen, and then nomenclature of the specimen. These things would be we should take care while working in starting our research work. So we need to make our voucher specimen, we need to identify this voucher specimen correctly, and we need to go with the correct nomenclature of the voucher specimen. Whether I am working for the taxonomy, whether I am working for, if I am working with the plant specimen, if I am handling the plant part, we should have our own voucher specimen. Then the importance, what is use of these herbarium sheets after your submission? So these herbarium sheets are basically compilation of knowledge of the flora of this one particular region, and they have lots of other they are the resource, they are the scientific resource. They can be resource for the anatomical studies, they can be resource for the pollen study, and or chemotaxonomy, the numerical taxonomy. They are the resource for the DNA barcoding, structure of the DNA. You can find out the even they are the reservoir for the as a gene pool. Okay. So how we should go? Suppose I want to identify the specimen, my own specimen, how I should start? So there are four different ways. Okay. One, I can take my specimen to expert who's already understanding these things, and so expert determination is required. So I will start one by one. I need to collect the specimen with the characteristics of flowers, fruit, roots. These things are required for the identification. So we need to recognize the specimen, and we need to compare the specimen. This is also important part of specimen comparison. We need to compare the specimen, our specimen, with with the herbarium. So there are already types are present in the herbarium. We need to compare. If our specimen is not matching with the herbarium specimen, with the type specimen, then I need to think again whether this particular taxon is valid, or this is a new taxon, or this taxon what I have collected, this is present in this particular area. Such questions we can ask and we can work further. And use of keys and taxonomic literature, which is very important. I will explain these keys and taxonomic literature afterwards. Okay, so we can which are these specimens which are submitted. So we should go for the expert determination, and expert determination, expert is going to put a label on this your herbarium sheet, and then you can this sheet is basically comment from the expert whether that particular specimen is like correctly identified or not. So so what is identification key? So identification keys is basically, it's a series of contrasting and contradictory statements. It's a series of contradictory statements. So so I will explain one by one. So look at this. So this is basically couplet. So what is, there are two different terms involved in this couplet and leads. So here you can see I have two contradictory statements like fruit with a keen fruit follicle. So these are there are two characteristics, they are contradicting to each other. This set of characteristics is known as the couplet. And each couplet, you need to accept one couplet, there's the statement, one statement as a lead, and then that particular lead is going to take you to the next level. So just think in this. So in this, suppose you have accepted fruit is akin and your specimen have akin kind of fruit, then you can go to the second character, calyx differentiate with corolla. And if this but they are matching, then you can, again you can reject or accept depend on the leads, and then you can accept one of the genus or genera. Though this is basically for the genus, and again same key will be available for the species also. So this is very much important, like step how we are going to read the keys. So you have the couplets. So in the keys also there are two different kind of keys. In front of you, you can see these are the indented kind of keys, and in case of indented kind of keys, they where couple leads are written not next to each other, you have various other leads in between. So between the couplets you have various other leads are also present. But in the brachiated keys, you will find each couplets are especially the written one above the other, okay, or in continuation form only. So one is basically fruit akin, fruit follicle. This is one couplet, and another couplet that tax. Then you have calyx differentiated, calyx not differentiated. This is another couplet. So you can read the keys, and you can suppose the fruit is follicle. In this, you can see the fruit is follicle. You can directly jump on the fifth number leads, and the spur is present, and your specimen is the delphinium. Like this, you can use the keys. Important. This is also important in the plant identification, the typification, and this is without type. Like your voucher specimens are also type. Your what whether you are working with taxonomy or you are not working with the taxonomy, what kind of specimens you are submitting to the herbarium, this is considered as a type. So what is type? So this type is basically a nomenclature, naming of the group determined by the type only. So type is a specimen which you are going to submit the or you have collected. So we will understand one and another different kind of type, yeah, one by one. So holotype. So what is holotype? When you are describing one specimen, suppose you got one new species, you have collected 10 different specimen of that new species, and now you want to describe it as a new species. So you need to select one type out of those 10 specimen. You need to select one type out of those 10 specimen as a holotype. and you are going to mention the number of the holotype, herbarium number, what author is giving in the manuscript, then only your specimen will consider as a holotype. And so you out of 10, you have selected one specimen as a holotype, and other nine are automatically becoming the isotype. So what is the concept of lectotype? Lectotype, when holotype is described, because everyone wanted to match the compare their specimen with holotype. So in the process, holotype may deteriorate. So after the holotype deterioration, the degradation, destruction of the holotype, you can make another lectotype out of the isotype. They basically, it's a collection from the original author who had described the specimen. I have one more example for the neotype. Lepidognathis bandrensis, it has the specimen was described by the Blatter, and from the bandrensis is basically location, Bandra location. They have collected this Acanthaceae member, Lepidognathis bandrensis, and Lepidognathis bandrensis is the specimen. So nowadays we don't find any collection from the Blatter. So we have though there is a no holotype, no isotype, and no lectotype. Then one fellow has, like Umesh Tiwari, he collected one specimen of the Lepidognathis bandrensis from the Dwarika region, Porbandar region, and then they have renamed it. They have made that specimen as a type for this taxon, Lepidognathis bandrensis. The such type is known as the neotype. Then another type is sen type. Sen types are basically where the type, there are various kind of, like there are certain types where there are many names have been given to the plants before this rule of typification came. So that time, that earlier authors have not made any type, like Linnaeus have not made any type, holotype. So for them, for the specimen of the Linnaeus, we can write the same type and then paratype. So what specimen, other specimen we are collecting from our region. Suppose I'm working on some physiology part, and I'm making a watcher specimen. My specimen will consider as a paratype. So this is the concept of the typification, and typification is important, and it will help as a reference material for other authors. So I will share one story. The Goa University professor Dr. Sanjappa came to the Blatter herbarium, and he have taken out the lots of the paratype from the, like students collection from the Blatter herbarium, and he, from those specimens he described the three new species. So this is the importance of the local collection, the paratype. Then another problem, as I have discussed earlier, the nomenclature part. So how to this is also important. How to understand this nomenclature part. So since ICBN is basically organization, so they have given 6 principles and various other codes. Around 90 plus codes are there. 93 codes are now. So they have various codes. Then 6 principles we can. So botanical nomenclature, principle number 1, I will read for you. Principle number 1, botanical nomenclature is independent of the geological and bacteriological nomenclature. So there is a so if they have botanical nomenclature have own the uh, principle. Okay, so it is independent. And the principle 2 states the taxonomic group determined by means of the time nomenclatural type. So if you are not making nomenclatural type, then your specimen is not, your work is not valid. Okay, so this is second principle. For every taxonomic group, you need to make a nomenclatural type. Principle 3, the whatever names are given by the people around the world, it will appear, you can accept the name only on basis of priority of publication, and there are other rules also, but priority of publication is important, and whoever is publishing first, that name will be accepted, uh, otherwise not. Then you have Then your scientific names, there should be Latin diagnosis of the scientific name. The principle fifth, there should be Latin diagnosis of the scientific name. The best example, the Lepidopteris ventrensis, why I spoke like just now. So Lepidopteris is the specimen was collected from the Bandra region, and they have given the name Bandrensis. And though they are converting the Bandra into the Latin words and for the scientific name, if they are using directly Bandra, it will not be acceptable. Even so, if you want honored, if wanted to honor one particular person, suppose I want to honor Dr. Kalam, so then the next yours you will write the name like Kalamai. Even there are some Mackin used to honor the bladder, so he used to like the specimen like Ceropegium blatteri, like this. They were using the Latin diagnosis. Even Chlorophytum borivalianum. So Chlorophytum borivalianum is Borivali is the location from where that particular the first specimen was collected. So they have converted the Borivali into Borivalianum. So like this, Latin diagnosis is important, and we need to convert the names English terms into Latin terms. Then earlier there was one rule: taxonomic publications should be in Latin. So now they have removed this rule. You can publish in English also. Earlier there was a rule, taxonomic manuscript for the taxonomic manuscript. Then last important principle, rule of nomenclature is are the retroactive unless expressly limited. It means the all this is going to be these rules are going to be applied on the before the whatever the names are there before also. So before the rule came, so this all principle is going to apply before publication, like Linnaeus all names also it is going to apply. Then you have certain other things I would like to share. So you have certain other things also like nomen conservanda. So nomen conservanda so you are going to write various things. So nomen conservanda is basically the nomen for particular conserved name. So what this nomen conservanda means? So nomen conservanda means like, so we are writing Fabaceae as well as Papilionaceae. If you have nom cons, so Fabaceae as well as Papilionaceae, both the names in the scientific literature, you will find both the names. So why is Fabaceae was priority? It was the published earlier, and Papilionaceae was published like later. Still, we are following the Fabaceae name Fabaceae. Why? And then because of this rule, nomen conservanda rule, Papilionaceae is the conserved name, and so we are going to write Papilionaceae everywhere. Then another nomen I would like to share, like nomen nudum. What is nomen nudum? So nomen nudum is basically the naked name, and naked name means whenever one particular scientist or author is going to describe his specimen, he has to describe his specimen uh, like all characteristics. Otherwise, without description, if he is publishing now, then his names will be illegitimate name. So this is the nomen nudum, and the nomen what I will share, nomen resectum. So nomen resectum also, you will find there are various places the name got rejected. So this is basically based on the code nomen rejectum. Then you have another, this is problem of the nomenclature problem I would like to share at the acacia. So you have two different kind of acacia on the screen, like acacia auriculiformis and acacia nilotica. There are two different kind of acacia on the screen. So so from the phylogenetic point of view, from the point of view of the origin, so this where phylod is present, so such kind of acacias belongs to the Australian origin, and where thorns are present, these acacias are from the point of view the African origin. So still there's a problem because both the specimen they belongs to the acacia, and so now the acacia type has changed. They have changed, therefore change. The type is the mimosa is a type for uh, the acacia region, and the specimen which has thorn we are calling it as Vauchelia, and specimen which have phyllodes we are calling is the acacia. Look at this. So how the plant identification is going to go to affect the other fields also. So this is the important, which is shocking thing we need to. So during the during the green revolution you will find the indigenous crops of the India. It was only rice varieties. There are one lakh 10,000 varieties of the rice were present in the India, and after the revolution only 6,000 now. This is the shocking data what the Hindu has published in 2012. And so we need to think. This is we need to think when the skills of the identification will disappear from the society, such kind of things will happen. So we need to one can take this challenge in the society, like identifying the rice varieties. How many rice varieties we have in India? Earlier this record is they are explaining there are one lakh 10,000 varieties are there, and now it's appearing up to 6,000. So how, whether really they have lost. So we need to think on these lines, and we need to identify them. And if and these collection will appear as a gene pool also for the future. Then another problem is the nomenclature of the cultivated plants. Cultivated plants, you will find there are lots of problems. So from the wild, because the floras and manuals, so they are incorporating only the wild plants. And for the cultivated plants, still new floras are started having, but older floras all you will find still they have not, or they have only wild plants. But the cultivated plants, this is also because we identification skills are involved in this, and the understanding the nomenclature is also involved in this. But there is separate nomenclature for the cultivated plants, the international code of nomenclature for cultivated plants. And these, so what others, like I want to quote what other scientists and authors are the writing about the International Code of Nomenclature for Cultivated Plants. These rules are are, are too complicated, and so there is the commercial sectors people, they are not able to use these rules. So this is also in the problem with the nomenclature of the cultivated varieties, and we need to understand this. Okay, so how to solve the problem of the nomenclature? Like we should have the plant taxonomic resources. And earlier we used to, like when I was learning this, so we used to have the hard copy of the index QNCs, and from that index QNCs, from one particular the genera, we used to find out the various references. And so like out of 400 names, we used to identify the name is the correct name and legitimate name. But now the work is easy. So we have the Q herbarium for the type, like which have the maximum type available on the online resource, and the plant list, and even the world checklist of the plant families. Even tropicos, they can help you under from the getting the references, and you can apply the nomenclature rules, and you can accept and reject. So for the so we need to make sure next time while writing manuscript we are going to write the accepted name, legitimate name. Otherwise uh, unlegitimate name and rejected name, synonym, these names are not valid names. So we need to understand this also. And so you in this is in this resource you will find out the synonyms also. What is synonyms? Synonyms is basically the not correct name for that particular taxon. So there, if the particular ranking and position is changed, then only that name is going goes under the synonym. So I will take the good example of uh, the mimosa. So your Acacia nilotica was described by the Linnaeus, and Linnaeus has described it as the Mimosa nilotica. So Mimosa nilotica he has described in 1753, and later the other authors came and they have changed the genus Mimosa to the Acacia. They have changed this genus Mimosa to the Acacia, and now it becomes Acacia nilotica. And now there are various authors they are treating that same Mimosa nilotica specimen as a Vicholia nilotica. So if you are writing the manuscript with the Mimosa nilotica, so this is not acceptable, and so it will create lots of confusion. So it is very much important to understand. So these So now use of computational methods. This is also important. Like because of the big artificial intelligence, what AI is coming, and still I'm in the support of AI. Like we should learn these programmings also as a part of like biology students. We should learn. So what they are doing, they are making various applications with the deep learning. Though what is deep learning? They are basically the training their software to identify the various pictures of the various color shades, and those after the training the softwares they will get your that particular application will get and correct, try to identify the specimen. But the problem, like you have Google Lens, so your Google Lens is going to, but problem is there are various from the external morphology you can do this, but there are various species which are segregating to each other from the internal morphology, and these applications do not have that data. So still we need to find out the solution in this. So with the use of computational method is not valid method for the writing manuscript. So I have identified the specimen from the Google Lens, these things will is not valid. And another thing, identification with the photograph, like I would like to make a serious comment here. So identification of the plants with the photograph is not the valid, like from the scientific perspective. If I want to write one so for general awareness this is different story, but if I wanted to write a manuscript, if I want to do a research work from that specimen, so identification from the like pictures is not valid. So I will share one story, like uh, mumbai university Mumbai University has rejected one thesis because that thesis uh, has the only photograph fellow has submitted for the checklist, though they have rejected that. So we have uh, so we should have the synonyms, we should have the citation author citations as well, the while writing the taxonomic literature. So only the pictures of the plants are not the part of the taxonomic literature, are not part of the references. Then so I so with this with this I would like to thanks everyone for the opportunity, this opportunity. And so I would like to thank you, Madam Arslan, Madam. So you have I, I tried to share things what I learned from the same department, Gujarat University department, and from my teachers. So if like my flow was not good, then it's I would like to have apology for that. And if some comments I made, still okay, madam.
Yes, Rupesh,
Thank you,
it was
sir.
nice. Thank you. Ha, go over, Dhruva. Yeah, please.
Thank you, sir. Thank you so much, sir, for delivering such an informative talk. On behalf of department, I thank Rupesh sir for sparing his valuable time. Now I request Bharat sir to deliver the formal vote of thanks.
I think there's some problem with the link. Yes. Yes, Bharat.
Yes, so sorry for this inconveniences.
It's
Just
okay.
about the link. Uh, Respected Professor Himanshu Pandya, Dr. Jagdish Pausar, Dr. P.M. Patel sir, Professor Parthiben Dave, Dr. Rupesh Maurya, my colleagues and dear participants. At the outset, I thank our Honorable Vice Chancellor, Professor Himanshu Pandya, for constant encouragement and guidance. I also heartily thank Dr. Jagdish Bhavsar sir for his motivation and help. I thank our Registrar sir, Dr. P.M. Patel, for his timely support. I thank our chief guest, Professor Parthiben Dave, for sparing her valuable time for us and give the inspirational and the motivational speech and about the knowledge regarding to this identifications and classification. I also thank our distinguished speaker, Dr. Rupesh Maurya, for giving an insight into plant identification and also content regarding to IUCN. I thank all the participants for their enthusiastic participation in today's webinar. I thank Dr. B.S. Agrawal sir, Director, Rolwala Computer Center, Gujarat University, to facilitate live streaming of the event on university Facebook page and YouTube. I thank Mr. Sagar Panchal from GUSEC for technical help. I also thank Ms. Dhruva Jani for anchoring this webinar so gracefully. And last but not the least, I thank my colleagues in the department for their timely help. Thank you. Thank you.
Thank you, sir.
Thank you,
Shakespeare said,
madam. Thank you, sir.
Yes.
Shakespeare said, what is in the name? But name is the most important attribute we can have. Today we have learned that every plant can be easily identified by following Sir's guidance. I once again thank you all for joining in the invited talk. The feedback link is on the screen. Please fill it up and get your e-certificates. Thank you. With this, we come to the end of the session. Thank you all for joining.
Thank you.